I'll answer the prompt. All right. So today I'm going to tell you about this group of Archaea that no one can pronounce that I fell in love with during my PhD and have not been able to stop studying. So I'll give a little background on what they are, why we care about them, and what I'm doing with them at the University of North Carolina in Wilmington, where I landed last January. So I lovingly call this the Wonder Archaea. I'll explain why in a little bit. So today I'll go through some basics. So we'll start by introducing non-extremophilic Archaea. I'll talk about nitrification, which is my favorite part of the nitrogen cycle, and the discovery of ammonia oxidizing Archaea, what makes them interesting to me, and then what keeps me studying them, why I haven't stopped since graduate school. And to the right is an image of the enrichment culture I worked with as a postdoc. This is Nitrous Archaeum limnium, and I think it's very photogenic. It's motile by a single flagella, which you can sort of see here. And then it has a crystalline S layer, so it kind of sparkles under the microscope. So it's really nice to show off. So we'll start with the non-extremophilic Archaea. So for those of you who don't study this domain of life as much as me, Archaea in general are one of the 3 domains of life. We have bacteria and eukaryotes, and then Archaea were established in the late 80s, early 90s by Carl Woese and his colleagues, who discovered through sequencing that this group of organisms that used to be called the archaebacteria were actually their own domain of life and distinct enough from bacteria to warrant that. My favorite anecdote to give my undergraduate students is that we as humans are more similar to pine trees than bacteria are to Archaea. So that kind of blows your mind. You don't think about that, but cellularly, bacteria and Archaea, even though they're single-celled and they may look the same, are totally different. So they're single-celled organisms. They don't have a nucleus. They're first viewed as extremophiles. So when you think of Archaea, I would be shocked if you said you didn't think of hot springs or salt lakes or something of that in mind. Now we find them globally, and it's sort of my personal mission to teach people that and to try to tell my friends who are high school teachers to teach their students that in the ocean behind us, in your garden soil, there are Archaea everywhere. So they're both similar and distinct from bacteria. I won't go into all of the details, but just a few examples. They have ether-based lipids instead of ester-linked lipids like bacteria have. Their transcription and translation machinery are very similar to eukaryotes versus bacteria. However, like bacteria, they have circular chromosomes and asexual reproduction. And so the group that I study specifically are the Thaumarchaeota. So nice job on the pronunciation. This comes from the Greek word thaumas, which means wonder, and archaios are ancient primeval. So this is where the wonder Archaea come from. And so within this tree of life, they fall here within the Crenarchaeota. And again, we think of these organisms primarily as extremophiles at first. So Archaea were first described in hot springs and salt ponds. And that was what we thought for a very long time, especially at the origin of Archaea. I guess I should say the non-extremophilic ones we knew about from the start were the methanogenic Archaea that's already studied early on. And so those can be found in marshes and elsewhere. They're in a different phylum within the Archaea that I'll mention. So the Thaumarchaeota are non-extremophilic Crenarchaeota. In the early 90s, Jed Furman and Ed DeLong's group, 2 separate studies, looked into the coastal ocean. And this was sort of the early days of 16S sequencing and doing environmental surveys of microbes. And they discovered that when they looked at the phylogeny, they looked at the relationships with these trees here, that they had groups that were related to Archaea and related to these extremophilic Archaea, but they were taken from the coastal ocean, 2 different parts of the country. So this made them start wondering, and I'll talk about sort of the history there. But now, like I said, we find them everywhere. So these are a few of the study sites that I love. But other places like the coastal, freshwater systems, soils, they're everywhere. And so a little bit closer into the Crenarchaeal phylogeny, just to introduce you to some of the major players within the Archaea that we think about. This is an early tree of this. So the main formerly kingdoms, phyla of Archaea that we think of are the Euryarchaeota and the Crenarchaeota, which are both in orange. We also have the core Archaeota and the nanoarchaeota that we're learning more about over time. And it's within this Crenarchaeota group that these marine group 1 low temp were first isolated. or first identified, and now we know them as the Thaumarchaea, and it's sort of this branch here. And if you've ever seen in the literature the abbreviation TAC, that is for Thaumarchaea, Eigarchaea, which aren't in this image, Crenarchaea, and Corarchaea, because they're all fairly closely related. Over time, with more sequencing, this distinction from Crenarchaea became more clear. So this is just one example of a tree where they took a bunch of genome trees. I'm sorry, the text is so small. This is like a tradition in all of my lab groups, is to show an unreadable phylogenetic tree as part of a presentation. So here we are. But basically highlighting at the time, in 2008, we only really had one genome of this group, but it was so distinct, here's Synarchaeum symbiosum, from the others, that they gave it its own phylum, and this is the paper that named it the Thaumarchaea. And so this is our first suggestion of it being an independent phylum. I will take a second to nerd out on taxonomy, because that's one of my favorite things. The advent of sequencing and making genomes out of metagenomes has really transformed the way we think about higher order ranks of microbes, and I think it's for the best, but it has led to a little bit of chaos. Because we are naming, creating, destroying, splitting, recombining phyla that we've known forever. And so my Thaumarchaea are no exception. So if you look in the literature, you can see them as marine group 1 Crenarchaea, Thaumarchaea, Nitrososphaerota, Thermoproteota. So I will keep calling them Thaumarchaea until someone tells me to stop. But currently, if you see it in any of your current sort of ribosomal databases, it will show up as phylum Thermoproteota, class Nitrososphaeria. And that's named after the first soil isolate. So we found these non-extremophilic Crenarchaea. What do they do? And so it's a spoiler because I've told you already that they're involved in the nitrogen cycle. So I'm going to introduce nitrification now and then get back to the discovery. So the nitrogen cycle, if you're not as familiar with it, it begins with nitrogen fixation. So we're in the oxic part of the ocean. I'm generally a marine focused person, so this is a marine nitrogen cycle. Nitrogen fixation brings nitrogen gas into the organic phase. This is basically in the form of ammonia. Nitrification then takes over and converts this through a few transformations to nitrite and then nitrate. Then this can go through a bunch of anoxic processes, including denitrification and anammox that serve to transform this back to nitrogen gas and release it into the atmosphere. There are other processes like DNRA that can also transform nitrogen and return ammonia to the pool, but this is the basic structure. So we're going to focus on the oxic parts because it is ammonia oxidation and nitrification. And one of the cool things about this process that I love and why my students all know nitrogen is my favorite cycle, because it's super microbially mediated, and it also, nitrogen goes through 8 electron transfers. So when we think of nitrogen, nitrification itself, it actually moves from the most reduced to the most oxidized forms of nitrogen. So ammonia on the right is the most reduced form that we see. Nitrate on the left is the most oxidized form, and that's a +5 state. And so, like I said, it spans 8 electron transforms. There's reactions to catalyze almost all of these intermediates that are within biology that's mediated by microbes. And so nitrification itself, ammonia oxidation, is this entire transformation. So ammonia to nitrite and then nitrite to nitrate. So it spans the whole length. And this is oxidation. So the 2 steps, we have ammonia oxidation and nitrite oxidation. And there's a hydroxylamine intermediate in the middle of ammonia oxidation. It's catalyzed by 2 organisms. And we've actually known about this process since 1890. When Winogradsky, if you've heard of Winogradsky columns, he actually was the first one to discover ammonia oxidation in bacteria and isolated some, and was able to show that it was a 2-step oxidation process and that his isolates could grow completely from CO2. And it didn't grow when they left out either ammonia or nitrite. So those are the first and second step of nitrification. And this is just a quote that he said, basically outlining that it's contradictory to how we think about physiology. At the time, we thought that it was only done by plants using light. And so this was starting basically the first evidence of chemolithoautotrophy, which we know now is a really important process and really dominant throughout the biosphere. But at the time, we only thought of phototrophy. So we've known about it for a while. So nitrification itself, now that we've introduced it, I said it was catalyzed by two different organisms. So we'll start with, we knew that there were ammonia oxidizing bacteria from Winogradsky, and we also know there are nitrite oxidizing bacteria. And if you're into a lot of physiology, Sari and I share a professor who taught us all about these types of things. But just to think about it, it's two steps, two organisms. So ammonia oxidation, this is the rate limiting step. It gets the most energy out of it. Like I said, there's a hydroxylamine intermediate, but this is 4 electron transfers. And then with nitrite oxidation, that's the second step. It gets you less energy. It requires the first one to occur. But basically, this maybe starts to give you a little bit of hint that the ecology between these organisms is a little different. I don't have enough time to discuss all the details about them. I'm happy to chat about them later, though. So there's ammonia oxidizing bacteria. There's also ammonia oxidizing archaea, and this was discovered earlier this millennia, at the beginning of the 2000s, so right before I started graduate school. And this was done with the isolation of this first organism, but I'll step through the history of that because we had some hints before. So back in the early 2000s, we started sequencing the ocean, and that started with this study out of the Venter Institute that generated a ton of sequences from the Sargasso Sea and led to a lot of really cool discoveries within microbiology, and one of which was a piece of this gene, this functional gene. And simultaneously, there was a group now in Vienna studying uncultivated archaea using phosmid libraries led by Christa Schleiper. And they also found a similar gene. And so these are the evidence from those two papers. So one is just a paragraph. It says that we found this functional gene, ammonium monooxygenase, that's involved in ammonia oxidation, but it was not bacterial. It seems to be archaeal in origin. And then in the Schleiper study on the right, this is their soil phosmid where they found an archaeal 16S on the same region as an archaeal AMO, so the ammonium monooxygenase. So this told them that this was possible. And so they were able to show that this was a different sequence. This is just an example. I had friends ask me all the time, well, how can you tell it apart from bacteria? We're lucky the primers can pick up. We can look at sequences and tree them together. They're very different. It makes ugly trees if you do that, but you can see this distinction. So highlighted in yellow, this is the thaumarchaeal functional gene, ammonium monooxygenase, or AMO, and we use the alpha subunit, so AMOA. And then in gray, we have the one from ammonia oxidizing bacteria, as well as PMOA. And this is because this is closely related to particulate methane monooxygenase. So we can tree all of that together and show that it's a distinct organism, a distinct gene. And now we use this in everything. So this is our single functional gene. If we're doing any surveys, quantitative PCR counts, doing just simple sequencing, this is what we use in addition to the 16S, but to study ammonia oxidation. So after the scientific community has found evidence of ammonia oxidation, it wasn't until they isolated one from a gravel aquarium in Seattle with Dave Stahl's group that they could prove that this did ammonia oxidation. And around the same time, my postdoc advisor, Chris Francis, did a study on this one gene, AMOA, and showed that it was found all over the world. So these studies together started to give us a hint that these organisms were important and possibly even more important than the ammonia oxidizing bacteria. We also have had other studies on the organisms, many more that I don't have time to go through completely, including some of mine, but just some other, basically these were the foundational studies in the proving that they existed and then why they might be more adapted to oligotrophic environments than bacteria. And so with this isolation of Nitrosopumulus maritimus, this is the first isolate of this group, they were able to show the production of nitrite, which is shown in these triangles here, matches the consumption of ammonia in squares, and this aligns with the cell growth so that they could grow exclusively from bicarbonate and ammonia And so they were chemolithic autotrophs. So this is our foundational proof that this works. So now we take our nitrogen cycle, our functional genes, and we add the Archaea. So both 2 domains of life do the ammonia oxidation, 1 does the nitrate oxidation. So since these foundational discoveries, I mentioned the sequencing survey by Chris Francis. We've seen them all over the world, and we now know that they're really high abundance. My PhD advisor has this lovely study from the Arctic Ocean where he talks about the abundance and diversity of ammonia oxidizing bacteria in 2002. Then ammonia oxidizing archaea were discovered, and we looked back, and basically they outnumber the bacteria 10 to 1,000-fold. So it's a great study, but now we know that the archaea are more important, and that tends to be true in most environments. High nutrient environments, less so, and agricultural soils, et cetera. But we'll focus on marine environments today. One of the coolest things I found about this group, in addition to them being everywhere and being fairly abundant, is that they have a really interesting distribution in the environment. So I'll call this a variable distribution. I'll give some examples with depth, latitude, and season. So if we look at their abundance in the ocean, this is from a study off the coast of Hawaii in the Pacific Ocean. And so we're looking at the counts of 2 different types of these archaeal genes. We have AMOA on the left, 16S on the right. Depth is on the y-axis. We're looking at abundance on the x-axis. And if I draw a line at 100 meters, what you'll see is that there's a significant fold change above and below the line. And so as you get deeper in the ocean, these organisms become much more abundant. And in some cases, we think of them as being 20 to 30% of all the single-celled organisms in the deep ocean. So a big contributor to biomass on Earth. By latitude, they're more abundant in polar regions than in other regions. And they show these really cool seasonal patterns. So this is from a really early survey in the late 90s, before we knew they existed, but as ammonia oxidizing archaea, but after we knew that they were non-extremophilic archaea in the environment. And so using some fish probes, Allison Murray did some counts, and what you can see already by the seasons is that there's a significant drop in abundance as it goes from winter to spring. And although in Sweden, you're probably quite familiar with this, when you hit winter, it gets pretty dark. And so when the light is off, they're really abundant in the surface ocean. When the light starts coming back on in spring, they basically disappear. And this, in fact, is coincident with around the same time, this look at UV radiation. And so they tend to not like light. We can debate a long time why that is. I have some work in that, photo inhibition, but basically changing seasons changes their abundance. And we think that's a really neat thing. So that's a preview to what I think makes them interesting, but I'll go through a few other examples here. So they're part of a coupled process. This is something that I think is really neat in the environment. They're really important for nutrient cycling, but also there's another group of organisms dependent on them. And for us to get this complete transformation from ammonia to nitrate, it requires these 2 organisms working together. So I'll talk about a little bit of my work here at the end, but basically the idea being if the coupling is going well, we're getting what we need to happen to nitrogen transformations in the environment. And if there's something disrupting it, we might not, and that could have some interesting consequences. They impact nutrients themselves. So I introduced the nitrogen cycle itself. This dictates the transformation of nitrogen species themselves, but this is part of a broader process of nutrient cycling. So I won't go through this entire figure, but this outlines a lot of the other processes in the ocean, including new production, respiration, photosynthesis, and nitrification sits right in the middle because it's transforming this ammonia, which is a really important nutrient, to nitrate, which can also be used, and both forms can be used by phytoplankton. We think of nitrate being used in new production. So it's a little interesting as we think about the generation of new production in the environment that this is actually also regenerated. It's not just, it's regenerated by a microbe. And in fact, most of the nitrate in the ocean was formed by nitrification happening over the history of Earth. So another thing, if you go out on a cruise and see nitrate, you can thank these ammonia oxidizers and probably the Archaea, because I bet they came first. They're also really good growing on ammonia. Ammonia distinct distinctly. We mentioned the cultures, which is shown here as well. In these low nutrient environments, they're really good at just growing on it and on it in really low concentrations. So on the right is a graph of the substrate affinity of this isolate, SCM1 from Nitrosopumulus maritimus in red. And this is one of the highest substrate affinities that was seen up to this point. On the right is a fungus, I believe, that had a really high, the highest known substrate affinity, and this was basically second best. So that just means that in the ocean, in any environment, if there's an ammonia molecule there, they can find it and they can make a living on it. So this works really nicely when we're studying the open ocean. We know that they're going to be there. It also makes culturing them interesting. I'll talk about that as well. But one of the best tips I got as a graduate student on how to cultivate them was to put them in a dark corner of the fridge and come back in 3 years, because basically they're the survivors. Everything around them is going to die. Their nutrients will be recycled and regenerated, and these archaea are just going to stick around, take whatever ammonia they can get, and chill, literally, and wait for things to get better. So I think it's a fun quality about them, and means that I just have a lab full of bottles of water waiting for these archaea to become the only ones in them. If you're interested in carbon cycling, they use a highly efficient carbon fixation pathway. There have been a number of studies over the years. It might be the most efficient energy-wise, but so to couple their ammonia oxidation to carbon fixation, this is through this 3-hydroxypropionate, 4-hydroxybutyrate pathway. It's a modified version of that that's found in other archaea, but basically this just helps them generate acetyl-CoA and biomass. And I'm not going to go through all the steps of it, but this is the pathway itself. There are some cool studies on the different modifications that Thaumarchaea have made over time. But we think of 2 key genes here, the acetyl-CoA carboxylase and hydroxybutyryl-CoA dehydratase. And these and other enzymes are being looked into as ways that we might think of transforming carbon in the biotechnology sphere, or perhaps for other purposes industrially, because it's so energy efficient. If you're interested in greenhouse gases, Thaumarchaea also produce nitrous oxide. So this was first shown, this is from Alison Santoro. This is now an enrichment of Nitrosopelagicus brevis. This is before they named it. But basically during regular ammonia oxidation, they produce nitrous oxide, and this was not known up until that point. And basically, so the graph on the left, we have time on the x-axis, concentration of N2O and nitrite on the y-axis. And basically in white are the nitrite, excuse me, and in black are the N2O. And so as they're producing nitrite, they're also producing N2O from ammonia. And N2O, just as a reminder, is a really potent greenhouse gas. It's about 300 times more potent than CO2. There's much less of it in the atmosphere, but it is being generated by this really abundant organism. And we're still trying to figure out exactly what makes it choose, or maybe not choose, but what might push it to produce more nitrous oxide versus nitrite. That's an active area of study. On the culture that I worked with, that I showed off with the pretty S layer, one of the former postdocs in my postdoc lab, Marie Lund, who's in Denmark, so near you all, was working with Nitrous archaeum limnium and studying N2O production during normal growth. And that's what this graph represents. The squares are N2O production. The bars on the chart are the abundance of archaea and bacteria, which there aren't very many bacteria because it is a co-culture, and the production of nitrite or ammonia, consumption of ammonia. And basically the cool thing that Marie found is that this N2O was produced in a way that matched their nitrite reductase gene, or NIRK. And so this became, we're leading to some clues that we're starting to rethink the nitrogen cycle. Like I said, N2O production was not previously known until we started as an important component. And we still don't fully know all of the components of ammonia oxidation for archaea. We don't know all the enzymes involved. And so this has led us to change our view of nitrification. And I won't go through all the details of these figures for time, but I'm happy to answer some questions about them later. But some of the new things that we found actually is that an enzyme like NIRK is perhaps critical to archaeal ammonia oxidation. In bacteria, they use a different enzyme pair. So they use the AMO enzyme that I told you about, and then hydroxylamine oxidoreductase, which is not found in Archaea. So we're still trying to identify what this second part of the process is. But as part of producing N2O, they also have a nitric oxide or NO intermediate. And we've thought at first that that was only present in Archaea, but have now discovered that it is an obligate intermediate in bacteria as well. So the discovery of the Archaea has transformed a process that we thought we knew everything about since 1890, that we maybe don't know everything about it. And the last word on this that you may have caught up is Comamox. And so since the discovery of these Archaea in the last few years, we've discovered that there are actually some nitrite oxidizing bacteria that picked up an AMO gene and are now able to completely oxidize ammonia. So instead of it being 2 steps, 2 organisms, it's 2 steps in 1 organism. We don't fully understand their diversity or importance in the global environment. They tend to not be super abundant in ocean systems, but it is something to look out for. And so now I'll go into some of my own personal interests, what keeps me studying them. I mentioned cultivation. I have bottles of these in my lab. These are all of the cultivated representatives we have so far. If there's a CA in front, it's an enrichment culture. And these come from a bunch of different environments, which I think is really important to our understanding. So it comes from marine sediment in the water column. We have a group from the low salinity freshwater, which includes nitrous archaeum that I have in my lab, Chris has in his lab in Stanford. We have some from sediment, and then a bunch from soils. And so, and even a few from hot springs. So I said they're non-extremophilic. There are some that are extremophilic, which really makes us wonder where they originated from. So we've been able to cultivate a ton of them, but there's still a lot that we have not cultivated and that we don't know a lot about. And one of my favorites of these is in the deep ocean. And so in this figure, this is a phylogenetic tree that's probably easier to read of groups of this functional gene that we use. And in purple stars are where we find all of the cultivated lineages. So there's a lot of groups that don't have them, including this glaring B group. And so to introduce this, and you'll see that I've marked out species. I think species can be a dirty word in microbiology. And we could debate for hours how you define a microbial species, as I'm sure you've heard about in many other talks. So in my group, we refer to them as ecotypes instead of species. It makes it easier. People aren't mad. But this is because it's a complicated concept. And up to this point, only in the last few years, we've started thinking of Thaumarchaea beyond being this huge phylum. So viewing it as one entity, telling you all these patterns that happen worldwide and all of these diverse habitats is not ideal. So instead of trying to argue which are species, we can look at ecologically relevant patterns and define subtypes within. So luckily with these groups, their phylogenetic distribution maps a lot of their ecological distributions, so we can use ecotypes to define. And just to prove that we're not making this up, cyanobacteria use ecotypes to define their highlight and low light classes. So I'll just introduce, because I think about them all the time, the major ecotypes that we study in the ocean. So this comes from these phylogenetic groups that match deep water and shallow water within that. And so these become our deep and shallow groups. And so like I was mentioning, there are no cultivated lineages from the deep water column B group. And so that's one of my personal missions to fix that. And then we have this whole ecotype as well that's really abundant, related to the first isolate. And I've done some work in Monterey Bay characterizing the abundance of all 3 of these. This leads to basically the driving question of my whole research program, which is, can we use these ecotypes to learn more about Thaumarchaea distribution, activity, et cetera? And that's sort of what I've been working on. And so throughout my career, I've been really lucky through graduate school and postdoc to study them in all of these different places around the world. I even made it into freshwater river systems as a postdoc, which is not something I ever thought I would do, but it was nice to cross the salinity divide and look at some interesting diversity in the western part of the United States. And just a few statements on things that I personally have found that I'm happy to answer questions about or talk about, or if you want to look these up later. So I found that they were sensitive to hydrogen peroxide. This is part of our debate over why they're sensitive to light, that perhaps reactive species may be the reason. I found that they bloom within estuaries along the southeastern coast, first in Georgia, that during the summer, when they shouldn't exist in the coastal ocean of the hot south of the United States, they're actually really abundant, and we're trying to figure out why that might be. I looked into some of their autotrophy, and other colleagues have as well, because they can take up urea and transform it. They have distinct patterns at species levels. So this is my ecotype work in Monterey Bay. And then I've been doing some cultivated work as the end of my postdoc and continuing it now, looking at their growth responses to non-optimal growth with the goal of trying to characterize unknown and undercharacterized genes. And so what am I doing now? I'm working here. The background behind me is a short walk from my office at the Center for Marine Science here at UNC Wilmington. We see an annual southern bloom here as well. So thankfully, my colleagues believed me when I guessed that I would find them here like I did in Georgia. So my PhD student, Parker Lawrence, and I characterized this bloom this summer, and we'll continue to follow up. And here we do see this decoupling of nitrification. So I hinted at that before. It's a two-step process. And what we see is that because these Archaea bloom, we have a buildup of this intermediate nitrite. And that persists for a really long time before it becomes transformed into nitrate. And so this seasonal decoupling of nitrification really impacts the nitrogen cycle. There's a ton of ammonia being consumed. There's a lot of nitrite being produced, which doesn't tend to stick around in the environment. And there's almost no nitrate being produced for an extended period of time, or not enough to be appreciable. And so this can impact phytoplankton because they use both of these and other organisms. And if the nitrogen cycle goes haywire, it can lead to things like eutrophication and harmful blooms. So this is why you should care about it, even if I haven't convinced you that they're one of the coolest organisms on the planet. And with that, I'll take questions. These are some of my favorite study sites, my favorite organism, and you can catch me on Twitter, send me an email. But thanks for the invitation again. Happy to chat and answer questions.